Hello and welcome to this webinar on the topic of SATB2 Genetics 101. This webinar has been made possible by a grant from the Child Neurology Foundation. I am joined this evening by Katie Bosanko, who received her genetic counseling degree from the University of Cincinnati in 2012. She currently works as a pediatric genetic counselor at Arkansas Children's Hospital and helps organize and run the quarterly SATB2 multidisciplinary clinic. Katie works directly with Dr. Zarate to build the SATB2 registry. So without further ado, I will turn it over to Katie for her presentation. Thank you for joining us, Katie.
Thank you so much for having me. So yeah, like she said, I'm Katie. I probably know a bunch of you guys just by working with you through the registry or through our multidisciplinary clinic that we have here. So thanks for joining, and I hope that you learned something new from this. So I think I will just jump right in. Y'all, I'm not great at Zoom, so I'm gonna do my best, but let me share my screen. And I have two screens, so that's why sometimes I'm looking away, but I'm trying to, okay. Can you see my slides? Okay, cool. All right, so the objectives of this talk is just to discuss basic genetics concepts and just the vocabulary that we use in genetics. I'm going to review different types of variants that can cause SAS. I'm going to talk a lot about how to read a genetic test result. So the letters and numbers that you see on a genetic test result, explaining a specific type of variant. I want you to learn a little bit of how to read that, if you will. And then also I'm going to discuss a little bit about just recurrence risk for parents, for future kiddos, and then also things that relate to siblings as well. Okay. Okay, I am going to start with the basics, guys. So for some of you guys, this is going to be review and maybe boring, and I'm sorry, but I want for maybe some people who don't know as much about genetics, I really want to start at the very beginning and work our way forward. And for those of you who already know this, this can be a good review. So, so this is just a good overview picture of genetics. And can you see my cursor? Okay, perfect. So our bodies, as you guys know, are made of lots of these microscopic cells. Inside our cells are our chromosomes. Okay, so our chromosomes look like this, and our chromosomes are just our DNA coiled together. Okay, so if we uncoil our DNA, it looks like this. So it's these little strands of DNA, and little chunks of DNA are called genes. Okay, we have about 20,000 different genes that are just spread around our DNA or spread around our chromosomes. And genes are made up of their own specific spelling of letters, if you will. These letters actually represent bases, is what we call them. And I'm not going to get into details of that, but these are the A, T, C, and G, adenine, thymine, guanine, and cytosine. It's just representing different molecules that make up our DNA. And a base pair is this pair of bases that comes together to make this double strand of DNA. So, so again, the T and A together, that's your base pair. But I like to think of genes as just a spelling of letters. And again, every gene has its own spelling of letters, because essentially what happens is the body reads that spelling of letters to then create a protein. Okay, a protein is actually what is produced and goes and travels around the body doing the different roles or functions that it's supposed to do. Okay, and a protein is made up of amino acids. So as you maybe learned in high school biology, amino acids are the amino acids are the building blocks of protein. So a protein is just a long strand of amino acids. Okay. So kind of backing up to chromosomes. So again, chromosomes is just our DNA kind of coiled up and packaged together. If we take one of our cells and pop it open and look at our chromosomes, this is what we would see. So in every cell in our body, we typically have 46 chromosomes. They come in pairs, numbered 1 to 22. And then we have a 23rd pair of chromosomes that are our sex chromosomes, telling us to be a boy or a girl. Boys have one X and one Y, and girls have two X's. The chromosomes 1 through 22 are what are called autosomes. So no matter if you're a male or a female, you have all of these chromosomes. The only thing that's different is that one pair of the sex chromosomes. Okay, so autosomes and then sex chromosomes. In every pair, we get one from mom and one from dad. That's how we have 50% of our genetic information from mom and 50% from dad. Now, the chromosome that we're talking about here is chromosome 2. That is where the SATB2 gene is located. So the SATB2 gene is on chromosome 2. So keep in mind then, we all have 2 copies of chromosome 2, which means everyone has 2 copies of the SATB2 gene. We get one from our mom and we get one from our dad. And so just kind of keep that in mind as we continue to talk. Okay. So this picture, here's chromosome 2. You see those little bands on chromosome 2. This is a picture that's just zoomed in on chromosome 2, okay? So you can see the little bands. And what we do is we use these letters and numbers as like an address on a chromosome, okay? So a chromosome has 2 arms. It has a shorter arm up here called a P arm, and then it has a longer arm on the bottom called a Q arm, okay? And then we have these numbers, again, to tell us a specific location. So the SATB2 gene is located at 2Q, so Q here, 33.1. And I know that's weird. We say 33.1 instead of 33.1. I don't really know why we do that, but that's our genetics lingo. So 2Q33.1, that's the location of the SATB2 gene. So within this little white band, that's where you can find the SATB2 gene. Okay. Okay. So the first thing I'm going to talk about are if someone is missing, either is missing or has an extra piece of this area of chromosome 2. So those are called deletions and duplications. A deletion is you're missing a piece of the chromosome. A duplication is you have an extra piece of the chromosome. There's 2 types of deletions and duplications. The first is what we call intragenic. So as you can kind of deduce, intra within the gene, intragenic. And so this person would have an extra or missing piece of chromosome 2 that is within the SATB2 gene, okay? So they are missing part of the SATB2 gene. And I will tell you, out of deletions and duplications, deletions is much more common than duplications. We do have a couple of people with duplications, but for the most part, I'm going to be referring to deletions because that's much more common. Okay, so here would be a test result. This would be what you would receive on your kid's test result. So it would say 2Q33.1 deletion. It would give you a lot of numbers. And then it would give you this other number. So lots of numbers. And then it might say it includes exon 9 and 10 of the SATB2 gene. Okay, let me break that apart. So we already talked about 2Q33.1 deletion. Okay, you're missing a part in that area. It's important to know what part is missing. And so that's where these numbers come into play. These numbers are the breakpoints for where the deletion starts and where it stops on chromosome 2. So we're talking about 200 million, and this is the base pairs. So this is saying 200,180,939 base pairs. So essentially what it means is on chromosome 2, if you kind of start at the beginning and you're counting all the base pairs on chromosome 2, when you get to 200 million, that's where the SATB2 gene is. It's way down on chromosome 2. And so this person is missing these base pairs between these two breakpoints. And so if you subtract those numbers, then what the number you're left with is 19.6 kb. So that means 19,600. So this person is missing 19,600 of those base pairs within the SATB2 gene. So 19.6, the kb means kilobases. So it's just a way of us making this number a little shorter. Instead of saying 19,600, we do 19.6 kb. Okay. I hope that makes sense. So again, breakpoints for the deletion and then size of the deletion. And then this part that says includes exon 9 and 10 of the SATB2 gene. So I'm going to go to another picture. So introns and exons, you may have heard these terms. So this is just a generic picture of introns and exons. So exons are what we think of as the more important part of a gene. So this is a gene, and exons are what are the most important. We call them the coding regions. They're the most important for producing the right protein, essentially. And then the introns, we're learning they're more and more important, but as far as making a protein, those introns actually get cut out of the DNA strand because they're not important in coding for that protein. And so, and that process of cutting out these introns is called splicing. So I see people trying to take notes. Tell me if I'm going too fast, guys. Okay, so splicing around each of these exons, there are what are called splice sites. It's just some letters, but essentially those letters tell the body where to cut out these introns. And so the body will cut out these introns at the splice sites, and then they push all those exons together, and then you have your modified DNA strand that is then read to create a protein. And actually it's a modified RNA strand. I'm not going to go into the difference between DNA and RNA, but it's a modified strand, and that is read to then create the protein. Okay, and later I'm going to talk more about protein synthesis and how that works, but I wanted to explain introns and exons and the splicing at this point. Okay, so back here, this person's SATB2 deletion included exon 9 and 10 of the SATB2 gene. So the SATB2 gene actually has 10 coding exons. They're numbered 3 to 12, just kind of weird, I get it. The 1 and 2 are just non-coding exons. So the coding exons are 3 to 12, and again, that would be a total of 10 exons. So again, this person is missing 9 and 10. They're missing a chunk, but not the whole gene. Okay. So that is intragenic deletions, and what all of that means. Okay, the next type of deletions and duplications is full gene deletions and duplications. These are larger deletions and duplications, and this is where they're missing the full SATB2 gene plus other genes that are around that area on chromosome 2. So for instance, this person's test result might say 2q33.1, q33.2. This is where the deletion starts, and this is where it stops. And then also the same as the first one. These are the breakpoints. The deletion starts here and deletion ends here. So this person, I pulled up chromosome 2 again. So this person has some missing pieces in this white 3.1 band, but also they're missing some pieces in the 3.2 band as well. So you can see that they just have a larger deletion. Okay, so again, it includes the SATB2 gene, but there are a lot of other genes in this region that they're missing as well. So when you kind of subtract these two numbers to figure out the size of this deletion is actually 5.7 MB. So not KB, MB. So this is millions of base pairs. So here, this person is missing 5,700,000 base pairs in their deletion. And I'm going to talk a little bit more about deletions later on as well. Okay, so those are kind of the beginning of the types of genetic changes that someone can have who has SAS. The next, I want to talk about point mutations or point variants. I will say point mutations, variants, and misspellings, all the same thing. I might use them interchangeably, and I'm sorry, but it all means the same thing. So again, just to review, we're now going to talk more about these, the letters that make up the DNA strand and the changes that can happen there. Okay, so I'm going to talk a little bit about making proteins, and this is important for you to fully understand the type of variant that your child has and the impact that it has on the SATB2 protein that then causes the symptoms that your child has. So, and I know I've already kind of started this conversation, but we have our DNA, the DNA is read, amino acids are joined together to create a protein. Once the amino acids are joined together, the protein then coils up and then again, goes through the body, traveling and doing the things that it's supposed to do. So the way that the DNA is read is over here. Okay, so up here you have your DNA template strand. And then you have your messenger RNA. That's kind of that modified strand that's already spliced, okay? And so that's what is actually read. And so every 3 letters on that strand is what is called a codon. And so your reading frame is 3 letters at a time, again, called a codon. And every specific codon correlates to a specific amino acid. So as the body reads 3 letters, a specific amino acid is then joined to that protein, to that amino acid strand or to that protein. So every codon is going to add an amino acid. The next codon is going to add an amino acid. The next codon is going to add an amino acid. So again, the letters are super important because that's what's telling the body the specific amino acids to add to that protein strand so that that protein is made properly so that it can then go and do the job that it's supposed to do. I hope that makes sense because I know that that can be tricky. But again, every 3 letters, amino acid. Also, one thing to note, because we'll talk about this as well, there's a specific codon or a specific 3 letters that tells the body when to stop reading the gene. So it's called a stop codon. So that is 3 letters saying we have all the amino acids that we need on this protein strand. We're good, we're done, stop. Okay, so that's a stop codon. That is natural. There is a stop codon for every gene, but the problem is when that stop codon happens prematurely, which we'll get to. So keep that in mind. Okay, so we're going to kind of come back to some of this, but I wanted to give an intro so that you can understand the different types of variants. So these are the different types of SATB2 variants or those point mutations that individuals can have. And really these types of mutations is not specific to kids with SAS. These types of mutations or variants, I mean, we see these on any gene. Again, we're talking about SAS, but these types of mutations are kind of universal. So there's 4 different types we'll talk about. There's missense variants. I have more slides on this, but because this is kind of an overview, but missense variants is where you have the whole amino acid strand, but one amino acid is changed. One amino acid is wrong, okay? All the other ones are what they're supposed to be, but one is different, okay? That's missense. Nonsense is where one amino acid changes and causes the protein to stop. So this results in the misspelling causes a premature stop codon. And so the protein stops forming and then it results in a truncated protein. Truncated protein means the protein is shorter than it should be. And if the protein is shorter than it should be, then it's not going to be able to work how it should. Frameshift variant is where there are extra or missing letters in the DNA. When an extra or missing letter is in the DNA, that causes the DNA to be different. So then the amino acid strand is going to be different. And typically frame shift variants lead to a truncated protein as well. And again, I'll get into it more. And then splice site variants is a variant that happens. So it's a mutation that happens at a splice site. So remember we talked about how the introns get cut out of the DNA strand. And so for this, the mutation happens at a splice site so that splicing isn't happening correctly. And so then that protein isn't going to function. It's not going to form properly, and it's not going to be able to function effectively. Okay. So again, back to missense variant. So this is what, if you're reading your child's test result, this is what it would look like for a missense variant. So here, this C is talking about our coding variant, so the change on the DNA. The P is referring to the change on the protein, okay? So essentially what this means is if you're counting the letters on the DNA, at letter number 346 of the DNA strand, typically we have a G, and instead this person has a C. Okay, so it is a simple little misspelling that happens. It changes the DNA code though, and so that's going to change the amino acids that are on the protein strand. So for missense, again, it only changes one amino acid. And so for this specific change, what this is saying is for the protein, we're talking about amino acid number 116. So typically that amino acid is a G or a glycine. And instead, because of this DNA change, that amino acid is an R or an arginine. So there's one amino acid that gets changed. All the other amino acids are what they should be or what we would expect. But because of this change, that results in the protein not functioning how it should. The protein is functioning differently, even with just one amino acid change. Now, other things that you'll see on the test result, heterozygous. So heterozygous means that this change was only found on one copy of the SATB2 gene. So again, remember I was saying everyone has 2 copies of the SATB2 gene. This change is on one copy of the SATB2 gene. And as we know, that is enough to cause SAS. Also, we hear a lot de novo. De novo means it was a new change in the kid. So that means that this change was found in a kid, and then parental samples were sent, so usually blood, maybe saliva or spit or something like that. And the parents were tested for that change, and their test was negative. They don't have the change, and so it's de novo, new in an individual. Okay, so that's missense variant. So here's another picture to kind of hit that home. So again, here's our spelling, C-A-T, C-A-T, C-A-T. So if this A is changed to a C, so just one letter change, then that's going to cause a different amino acid to be added to the strand. It's a different codon. Okay, so that is how the missense variants work. So this is a graphic of all of the missense. So this is specific to SAS. So here's our SATB2 protein, and these are all the different missense variants that we know about. So they've either been published by other people, or they are variants that we know about from parents sending us test results, or people who are in the SAS registry. And so so you can see here, some of these variants are unique to individuals, and other variants are seen in a bunch of different people. So like this variant is the most common, and this specific variant's been seen in 20 individuals with SAS, this one in 9, this one in 6. So there are some variants that are what we would call common variants, so that can happen more frequently. Now, interestingly, with missense variants, what we're seeing is that most missense variants are happening in certain areas of the protein. So they're happening in these green, what we call domains. So you can see they're kind of clustered here and clustered here. These green areas, these domains are really important for the gene to function how it should. These are binding domains. And so it's important for the protein to be able to do all that it's supposed to do to have these little areas working properly. And so as you can imagine, if there is a misspelling or a mutation that happens in these areas, if that domain isn't able to work how it should, then that's what's causing kids to have the symptoms of SAS. And so what we're seeing is, and we're kind of starting the process of doing a more official study, but what we're seeing is that having variants in these regions, missense variants in these regions, kids have more of the classic, what we think of classic features of SAS. If they have a variant that falls outside of these regions, so in these regions or these regions outside of the green regions, we're seeing that individuals might be a little bit more mildly affected, okay? May have less significant symptoms than individuals in the green area. And I'm not saying that means that we can predict what symptoms a kid is gonna have, because we can't. But what we want to look more into is quantifying somehow some kind of score of, mild to moderate to severe to show that these variants that cluster here seem to have, a more classic SAS picture. So more to come, but we think it's interesting, especially for those of you who have kids with the missense variants. Okay, I will move on. So the next variant is the nonsense variant. So here, same as before of how this is kind of set up on a test report. So what this means is, as you're reading, counting or reading the letters, at letter number 847, typically we have a C and instead this person has a T. That's their misspelling. What happens is on the protein level is typically at amino acid 283, typically there is an arginine. R stands for arginine, so that's the amino acid. But instead, this change has told the gene to stop. It has created a premature stop codon. So the X means that no amino acid is there. It told the body to stop forming that protein. So from at this point forward, there will be no more protein. And so that results, like I was saying earlier, in a truncated protein. Same thing with heterozygous and de novo, as we've already gone over. So again, to hit the point home, here are your codons and your amino acids. If you have a misspelling that causes the codon to be a stop codon, then the protein will stop being created. Okay. Frame shift variants are similar. So frame shift variant, so here's how you read this. So at letter number 1945, you have a dupe T. That means duplication T. So that person has an extra letter T in that location. So they just have an extra letter that has been inserted in there. They have all the letters before and all the letters after. They just have an extra letter. As you can imagine, if you have an extra letter that's inserted into the DNA, the reading frame is going to be shifted from there on out. So, because again, we're reading by 3 letters at a time. So if you're inserting a letter, it's shifting everything else. That's what frame shift means. It's shifting all the letters after it. So all the codons after that is going to put a different amino acid in, because that codon is going to read differently. So for this, at amino acid 649, typically we have an S, so a phenylalanine. So that a serine, S is serine. That has been switched because of adding the T to a F, which is phenylalanine. And then this says FS, frame shift, X40. So what that means is, because of this extra T, they were able to add on 40 amino acids after that, but at the 40th one, there was a stop codon and then the protein stopped being formed. So whenever you have a frame shift variant, sometimes some amino acids can get added onto the strand. It might be the wrong amino acid, but it can get added onto the strand. But at some point, for the most part, you're going to end up, a stop codon is going to be is going to come up prematurely, and then still you're going to end up in a truncated protein. Okay. So for nonsense and frame shift variants, both of those end up with a truncated protein. Okay. So again, just to kind of hit home here. So here we have our strand of letters. An A has been, so they have a dupe A. So they have an extra A here. So that's going to push all these other CATs over. So instead of reading a CAT, you're now reading ACA and then TCA and then TCA. And so then you're going to have a different amino acid strand from that point on. And then again, eventually it'll lead to a stop. So this is the graphic of the nonsense and frame shift variants that we know of. The red are the frame shift variants and the orange are the nonsense variants. So as you can see, these variants can happen all along the protein, okay? So, and again, this is the protein. These numbers are for the amino acid number. And so this can happen all along the protein. Now, one question we get very frequently, and it's a great question, is if someone has a truncating variant here at amino acid 100 versus someone who has a truncating variant over here at amino acid, let's say 500, does that affect severity based on how, if you're producing a smaller protein or a larger protein, but still isn't the whole protein? And really this has no effect on severity, okay? So no matter where the truncation happens, it still creates a protein that isn't able to function properly and can still cause any and all symptoms related to SAS. So there's no way for us to predict if someone would be milder or more significant based on where the protein truncation happens. Okay, and the last one is a splice site variant. So what this means is at letter number 254, around that letter is where a splice site is. So whenever you see a minus here, a minus 1 or 2, or a plus, it also can say plus 1 or 2, that means that it's a splice site variant, that there's a letter change in that splice site. And so here, typically we have a C in that spot, and instead this person has a T. That's going to alter that splice site, so splicing isn't going to be able to happen properly, and then that's going to end up creating an abnormal protein product. So here are the splice variants. These are less common, but here you can see these are our exons here, and so these splice site variants are happening right around where those exons end and begin, because they're happening right at those splice sites. Okay, so a few things to add about these different variants. So variants, as you can see from the graphics, variants have been found in all of the coding exons. Again, remember exons 3 to 12, those 10 exons, but half are located in exons 9 and 10. So that definitely seems to be more of the hot spot for changes in this gene. And then here, this is a paper that we published last year, just on as a mutation update paper. And so here you can see the different variants that we know of. Missense variants are the most common that we know of, but really, since nonsense and frame shift variants do the same thing, they result in a truncated protein, we would say that the most common type of variant is a protein truncating variant that causes SAS. You can see that splicing variants are less common. And then here are your intragenic deletions, so deletions within the gene, and those are more common. This doesn't include those larger deletions. We're actually in the process of publishing a paper right now that's focused on those larger multi-gene deletions. Okay. One thing that people ask, and it's a great question, is about genotype-phenotype correlations. And I've talked about this a little bit, but this is a genetics term, genotype-phenotype correlations, and that just, genotype means a specific genetic change. That's your genotype, the genetic change. Phenotype is an individual's symptoms. And so a hot topic in genetics in general is what kind of genotype phenotype correlations can we find? Can we figure out if someone has a specific genotype, can that predict what their phenotype will be? And so there's a lot of stuff going on. And honestly, at least from my perspective, it's hard to find these types of correlations. But within our SAS community and cohort, we are seeing some genotype phenotype correlations, which I'll get to in the next slide. But I just want to say, always remember that there is what we call variable expressivity, that individuals, even with the exact same genetic change, can have different symptoms. That is with any genetic condition, including SAS. And you've seen, even with what you know about this condition, you've seen the variability between individuals, but you can see that even individuals with the exact same change. I know it's fun to find other individuals who have the same change as your kids' change, you know, I mean, and I think that's cool, but that when you see that, you will still see variability within those different individuals. So just always remember that. There's no way we would ever be able to predict this kid has this change and that means they're going to have these symptoms, because we know even in the same family, you know, for genetic conditions that can be in the same family, we can even see people with different symptoms. So we're never going to be able to predict that fully. Okay, so a few things about intragenic deletions. These kind of the way they act is similar to like nonsense or frameshift variants. So those intragenic deletions result in a truncated protein, which then causes the signs and symptoms that we see. Larger deletions, again, in the paper that we're working on now, we do see that they have more growth problems with the larger deletions. And then they also can have other problems that are specific to some of the genes that are missing. So here is our graphic for deletion. So this is 50 people with one of these larger deletions. And you can see here, their deletion is in red. So all of these people, and this person's only missing just part of it. I don't know about this one. I need to ask Dr. Sharate. But anyway, but these people are missing the SATB2 gene, or at least part of it. But if they are missing this COL3A1 or COL5A2 gene, so some of them you can see are missing that because it's a gene in that area, we know that if they're missing that gene, that person is at an increased risk for aortic dilation. The aorta is the part of the heart that pumps the blood from the heart to the rest of the body, and that aorta can become dilated. So those people are going to require a lot more cardiac management, so monitoring and management. And then also if people are missing this BMPR2 gene, then that's a gene that's associated with pulmonary hypertension. And so there's going to be an increased risk for pulmonary hypertension. So in general, having a larger deletion doesn't necessarily mean that someone's going to be more severe or less severe or something like that. We definitely can still see a wide range in symptoms, but again, having these specific genes involved can definitely increase the risk for other types of symptoms. Okay, other genotype-phenotype correlations that we've seen that has to do with the different variants. So for nonsense variants, this category of individuals tend to have the lowest number of nonverbal individuals after 4 years of age. So 8 out of 29 were nonverbal after the age of 4, which is a lower number, significantly lower number than for the other variants. So maybe are a little bit more verbal. Also for nonsense variants, they're less likely to have clinical seizures. Now for missense variants, they tended to have the highest number of nonverbal individuals after the age of 4. So 20 out of 39 or 51%. And then they were less likely to have a cleft palate, and then also more likely to have seizures. And then for frame shift, the only thing found was they were just more likely to have feeding difficulties, which we know that's pretty common no matter what. But 96% of them had feeding difficulties. So nothing too earth-shattering here, but definitely some statistically significant differences, and we'll kind of see how that changes over time. Okay, I'm going to switch gears for the rest of this and talk a little bit about inheritance, mosaicism, things like that. I know that y'all have heard those, probably heard those terms before. So the first thing I want to mention is just the term autosomal dominant, because SAS is what we call an autosomal dominant condition. What that means is autosomal means the autosomes, remember we talking about chromosomes 1 through 22 are autosomes. So autosomal just means that this gene is located on an autosome, chromosome 2. And then dominant means that you only need one SATB2 gene to have a change in it to have symptoms. So recessive conditions, you have to have both of those genes have to be non-working, but for a dominant condition, only one gene has to be non-working to have the condition. Also, we use terms de novo versus inherited, which I was talking about de novo earlier. So one thing that some families ask is, what do you mean this wasn't inherited? This is a genetic change. So clearly, these changes are genetic. It happens in the person's genetics, but you can have a genetic change that isn't inherited. Some changes are inherited from a parent and some changes are brand new in an individual. Mosaicism is a term that means that someone has, some of their cells have a genetic change and some don't. So let me kind of show you what that means with this picture. So what happens with mosaicism is when early on, when an embryo is created or a zygote, so that's the fertilized egg is called the zygote. So very early on, that zygote does not have a genetic change, but over time, a mutation happens in a cell. And so that means from then on, from that cell, any of the daughter cells that are made from that cell are going to have that mutation. But then the other cells that don't have the mutation will continue to make other cells without the mutation. And so how that can result in someone having mosaicism, where some of their genetics, some of their cells have the genetic change and some of their cells don't. Okay. So we know that mosaicism can happen about 1 to 2% of the time. What that means is, so can happen several different ways. One of the ways that we've seen it is a parent can be detected to have what we call low-level mosaicism on the kid's test. So like a lot of people are diagnosed with SAS via whole exome sequencing. And so as I was mentioning earlier, parents for that test normally submit their samples, blood samples or saliva or cheek cell. And so sometimes what we find is that a parent can have a low level of mosaicism. Some of their blood cells had the genetic change and most didn't. So typically if we see that there's like 2 to 4% mosaicism. I mean, we're talking about a small number of cells that have the genetic change. So a parent wouldn't have SAS because 4% of their cells have the change. So they wouldn't have SAS, but if some of their cells have the change, then there's a risk that they could have a kid with SAS, because we don't know that mosaicism could be in some of the egg cells or some of the sperm cells. And so if that's the case in a parent who is mosaic, if it's in some of their egg cells or some of their sperm cells, then there's that risk to then have a baby that inherits one of those changes. So sometimes that mosaicism can be detected in blood at a low level like that. Other times it can't be. If the mosaicism is in a super low level, 1% of the cells has it, there's going to be no way that that gets detected on a genetic test. Now, it's tricky because if someone has mosaicism, it's very difficult for us to know how to counsel on recurrence risk, because we don't know how many of the egg cells or how many of the sperm cells have that genetic change. So really, if someone has mosaicism, the recurrence risk to have another baby with this condition could be up to 50%. Now, the other side of that is germline mosaicism. So this is where we do genetic testing, a child's found to have a change, and both tests for the parents were negative. Again, that's when we would say that the change is de novo in that individual. But there is a phenomenon called germline mosaicism, where that mosaicism only happens in some of the germ cells. So in some of the egg cells or some of the sperm cells, it's confined there. So the test would be negative. The parental testing would be negative because we're not testing their egg cells and sperm cells. But there could be an increased risk to have another child who inherits a change and who has SAS. So we say there's about a 1 to 2% risk for that to happen, for germline mosaicism. So when you are, in the Facebook group and you're hearing of families who have multiple kids with SAS, this is why. Either one of the parents has a low level mosaicism that we can detect, or there's germline mosaicism that there's no way of detecting. So when we talk about recurrence risk for this condition, we can never say it's 0%. We would always say it's about a 1 to 2% risk to have another child with SAS because of the possibility of mosaicism. And again, within our data that we have, it's about 1 to 2% of families who have at least who have another child with SAS that confirms that mosaicism. Okay. And then also I wanted to speak to unaffected siblings because I get this question a lot. So if a parent does have mosaicism, so whether we can detect it or not, if they have mosaicism, each kid either inherits a SATB2 variant or doesn't. So for that person's offspring, that kid has SAS or they don't. Okay. So if the child is not affected, they don't have the SATB2 variant. And so at that point, then that unaffected sibling is not at an increased risk to have a child with SAS. They're back to the general population risk. Okay. It doesn't skip generations. That child wouldn't have mosaicism. You either have it or you don't for the kids. They wouldn't have an increased risk to have mosaicism. Okay. My last slide. So someone earlier was asking about pre-implantation genetic diagnosis. I'm not going to go into this in great detail because I am not a prenatal genetic counselor and this is not my expertise. But some families do ask about, I have a kid with SAS, I want to have another child, what are my options? And so pre-implantation genetic diagnosis is one of those options. And what that is doing genetic testing on an embryo to screen for a genetic mutation or a condition. And then that process all happens via IVF. And then they test the embryo, whatever embryo is unaffected, then that's what's implanted into the uterus for a pregnancy to happen. And so what they do is they create the embryo outside of the body. So they fertilize the egg outside of the body, and then they're able to they let it grow just a little bit, and then they take some of the early cells from the embryo, they do genetic testing on those, and then, as I said, whichever embryo doesn't have the change, then they implant into the mom for a pregnancy. Overall, as far as PGD goes, it's pretty accurate. I was reading 98 to 100% of the time it's successful. And at this point, studies show that it doesn't lead to an increased risk of problems for the baby. Now, IVF in general, there are some increased risks for just IVF in general. And what we think is the risk for IVF is for methylation errors. So methylation is kind of a weird thing that happens with our DNA, where our DNA kind of gets stamped in different ways. And so the DNA will stamp, it kind of is telling the body which gene or area is from the mom and which came from dad. And so it's stamped to kind of tell the body that. And in some areas that's important for, the area to be there that came from dad, or the area to be there that came from mom. And so with IVF, there's a bit of an increased risk for methylation errors to happen. It can just throw off that stamping. And so that can lead to some genetic conditions. But as far as like genetic mutations, it's not thought to have an increased risk on new genetic mutations. As far as PGD within the SAS population and community, we have not studied that. I know of one family who has gone through it and it was successful, but we just haven't looked into it and we haven't asked a ton of questions about it. So that is honestly all that I know on PGD. So yeah, but it is an option. I know families have used it and it's been very useful, and definitely something to explore if you're interested. Okay, I just talked for 50 minutes. I am done. So please ask some questions. I think people were maybe chatting some questions, but I couldn't focus on talking and trying to look at this chat thing at the same time. Like I said, I'm really not good at Zoom.
Katie, the question that came through the chat was during the miscent variant slide. She asked, where does the first green domain start?
Hold on a sec. Can y'all see my slides?
Not.
I know. I don't know where they went. Where did they go? Hold on. Because I was going to go back to it. Here we go. Okay. Say that again. Missense?
Yeah, I believe that it came up when we were talking about the missense variant, and you had the slide about the green, and you were saying in the green section was where you were seeing more.
What number? Like what number amino acids?
Yeah, she asked, where can I ask where the first green domain starts?
Yeah, well, that's a good question. It's around 360, around 350 maybe. That's a guesstimate. I'm sorry, I don't know the answer other than that.
I had a couple questions that came through my email. So is SAS degenerative in nature?
Oh, as far as someone gets worse over time, no, it isn't. Not that we've seen. Now, remember, we're very limited in the adult population in general. So we don't have a lot of information about the adult population, but we are doing a study currently that is trying to explore that. And from what we've seen, and again, we'll work on getting the data published, but from what we've seen, it doesn't seem to be a degenerative thing. So we're not seeing progression in symptoms as meaning that someone's getting worse over time. Things like that. I mean, it seems pretty stable. I mean, everyone's going to have new things that pop up, but it seems pretty stable overall.
Great. The next question is SATB2 related to bone cancer?
Nope. Not that we know of.
Great.
That has not been shown in our data, no.
The last question I had in my my inbox was, does the nature of the genetic change, missense, nonsense, duplication, or deletion correlate to onset of symptoms or prognosis?
Right. No. So I kind of think I answered some of that, right? As far as the genotype phenotype thing.
You did. I just.
Yeah. Okay. Yeah. No. Yeah. No. At this point, there's nothing where you can say someone has this change and they're going to have X, Y, and Z symptoms, or they're going to be milder or more severe. Other than what I was saying with the missense variants that we're going to kind of be looking into, but yeah, nothing that can really predict or give us a specific prognosis.
So just to go through some of the questions that are on the chat thread, one person was wondering that the exact same mutation can cause different MRI scans, and they understand that there's some variability from child to child, but I guess their MRI scan showed hypomyelination versus a normal MRI scan. Do you have any comment on the myelination issue for the kids?
As far as type of variant?
That's a good question. Have you noticed that, I guess, first of all, is that common across, have you noticed that is a common thread for children with SATB2? And then also, do you see it in any one situation more than another?
Gotcha. Yes, seeing hypomyelination is a more common finding. I do not have numbers like statistics on the top of my head about that, but yes, we do see that pretty commonly, hypomyelination. But I don't think there's been any correlation as far as seeing hypomyelination more frequently due to one specific type of variant. But yes, definitely not uncommon to see.
Another says, my daughter's test results say novel variant with dad being a mosaic. Does that mean she is de novo?
So for that, novel variant means that that child has a variant that's never been reported before. So never been reported in the literature before. That doesn't necessarily mean that no one else has it, but it's just never been reported before. So that's what novel variant means. So if the dad is mosaic, I'm guessing it's a couple of percentage. I don't know if it has categorized the percentage for you or quantified that. But at that point, I would say it is inherited. So a dad has a low level mosaicism. So they have some tissue or some cells that have the change. And so then that's what got passed to the child. So that change actually started in the dad. It was a new mutation in the dad.
Any relation to glaucoma in SATB2?
No, not that we've seen in our literature. Not meaning that no one has had glaucoma, but it doesn't seem to be an increased risk for glaucoma.
Okay. Stem cell gene therapy for our syndrome, is that only for prenatal, fetal, or is there any application for after birth?
Yeah, to do like a stem cell transplant.
Something
like that. So, yeah, so, I mean, just doing a stem cell like at this point, doing a stem cell transplant, we don't feel like is a treatment. I mean, it's not going to cure things. It's not, I mean, it's not standard of care. I don't know what will happen in the future, but at this point, for most genetic conditions, you can't just do a stem cell transplant and not change things. So a lot of, there are some genetic conditions now where there's some new treatment that is able to go and change a gene or something like that, and that can help, but that's very rare, and we don't have anything like that for SAS. Now we do have Dr. Zarate is working on creating stem cells from individuals with SAS. So we got skin samples from individuals and their parents with SAS. And from that, we can actually create stem cells that have the SATB2 change in them. And then from that, you can create different tissue types from those stem cells. This is all in the lab, but you can create different tissue types from those stem cells. And then once you have a different tissue type, then you can start experimenting in the lab with different medications or different treatments to see if there's any kind of impact on those stem cells, to then see if there's anything promising that could actually help in real people. So we're at the very beginning stages of that, but the stem cells have been created, right, Allison? And the next step is getting them to where they need to go to start doing some experiments on them. So that's what I know as far as stem cells.
Allison, did you have a comment on that, or do we want to save that for another?
No, I was going to say that was a big project that we funded through the foundation last year, and they're in their final stages, so we can start pushing that out more.
Yeah.
There are a couple more questions on here. Is germline mosaicism the same as gonadal mosaicism? Is that interchangeable? And what age do you see seizures starting when they develop?
What age do you see seizures?
I'm sorry, yes, do seizures start?
I don't have numbers for seizures. We did do a seizure, SATB2 and seizure paper last year. I'm happy to share that with anyone if you want to send me your email address. I can't remember the age of onset. I mean, it can be any time, but I can't remember the specific age of onset. But yeah, like I said, we did do a paper. I'm happy to share it with anyone who's interested.
How about cases registered in older individuals? Is it a new, is SATB2 a new genetic syndrome, or are we seeing more cases in older individuals the more we test?
Mm-hmm. Yeah, that's a good question. Yeah, no, so our study that we're doing right now with the adult population, so we're doing some adolescents too, but from age 18 and over, I think we were able to enroll 23 people. So I think the oldest being 39, I want to say. So I was actually really pleased with those numbers, but I don't think it's that SAS is a new thing. I don't think it's that there are more kids with SAS than adults. I think that it is that more kids are being tested or are having genetic testing. The genetic testing that we do now was not around even 15 years ago. So whole exome sequencing, which is probably what most of your kids have had, that when I got out of school 8.5 years ago, whole exome sequencing was a new thing. So genetic testing is growing and changing very rapidly. And so a lot of individuals who are older, I mean, this testing was not around 40 years ago. And so, and then I think at least what I see in clinic is, as individuals get older, parents are just used to who they are, which is awesome. They've adjusted to life with them and they have a plan, and having a genetic diagnosis isn't going to change what they're doing. And so I think that there's a little bit less motivation because they're like, it's not going to change anything about our life. And also insurance changes. So a lot of times you can get better coverage for things in kids than you can in adults. And so it's just not, and then also genetic services are a lot harder to come by for adults than they are for children as well. So I think there's a lot of variables there, but I definitely don't think that it's less common in the adult population. We just don't know about as many adults.
And then can you define what a pathogenic variant is?
Oh yeah. Sorry, I didn't talk about that. Pathogenic. Okay, that's good. Yeah, thank you for bringing that up. So pathogenic variant is a variant that's disease causing. So it is a variant that we feel like, yes, this causes the protein to not function how it should. So most of the variants that your kids have are probably pathogenic. Sometimes we see variants that are called variants of uncertain significance. So you might see that on a test result. That means that we don't know. It might be pathogenic or it might not be. We don't have enough information for that specific change. We don't have enough information to say if we think that that could be disease causing or not. Most of the time in this population, we see a pathogenic or likely pathogenic variant. Every now and then we might see a variant of uncertain significance. Great question. Sorry, I should have brought that up.
Can you give us just a little background about what the role of the registry for SATB2 is and its importance for the foundation?
Yeah, so for those of you who don't know, the registry is just a compilation of clinical and genetic information and developmental information on individuals with SAS. That really all of the research that we do comes out of the registry. So the registry is our cohort of people that we are using to learn about the condition, to even learn what studies to do next, what things to look into more. And so, I mean, that's so many of like the images that I showed here are because of the information that we get out of the registry. So from a clinical standpoint, and really Dr. Zarate is, I think, the only one doing clinical research for SAS. I mean, that's how we've been able to learn most of the clinical information that we've learned over time is through participation in the registry. So it definitely, I know you register and you give us information and then you don't get much from it maybe, but really in the long run, that's how we're learning more about your kid's condition. So super important. And if you're not in it, I would encourage you to take part.
And one last question, and then we'll let everyone go for the evening. And someone was wondering about gastroparesis. Is that common with the syndrome?
I don't have numbers on that as well. I definitely have seen individuals with gastroparesis. I think we tend to lump GI issues just together, honestly. So I don't know if we've teased it apart necessarily with gastroparesis, but we definitely see GI things, constipation, reflux, gastroparesis. But again, I think we probably lump those together.
Great. Well, Thank you, Katie, for joining us. Thank you for helping to break down this extremely complicated topic into slightly more manageable parts, and just being available and everything that you do for our foundation. It means a lot to our families. And so we will sign off, but thank you again, and hope everyone has a very good holiday season.
Yeah, thank you. I hope that was helpful. Feel free to email me. Let me, hold on. Are my slides still up?
No, they're not, Katie.
All right, let me, because someone was asking for my email address. I'm going to share my screen again, and you can see my email address. Can you see my email address now?
Yes.
Okay. Yeah, so feel free to shoot me questions. I work with Dr. Zarate. If there's questions I need to ask him, I'm happy to. We love to be a resource for you guys. And we're always happy to help however we can.
All right. Thank you very much.
Thank you, guys.